Hello and welcome to the webinar. In this webinar, we will highlight the latest advances of circRNA in cancer. circRNAs have been attracting more and more attention lately, with rapid growth in circRNA research and PubMed publications. Many new circRNA biological and clinical functions are continually discovered. In particular, circRNAs play important roles in many types of cancers. Their unique circular form has special properties in biofluid cancer biomarker use. circRNAs can be epitranscriptomically modified, which has been found as a new critical player in gene regulation. circRNAs are an unusual class of RNAs. Many functions have been discovered for circRNAs. circRNAs are well known for their microRNA binding capabilities as microRNA sponges to regulate silencing complex-mediated gene regulation. Likewise, circRNAs can bind RNA binding proteins as RBP sponges. There are several hundred RBPs performing a variety of biological functions, such as transcription, splicing, RNA processing, RNA transport, RNA localization, RNA translation, RNA stability, and RNA modification. Over the years, circRNAs have been found to be involved in all these RBP-related functions. circRNAs can also be the guiding scaffold for proteins complex assembly. Many circRNA and protein interactions have been cataloged in circInteractome database. Although circRNAs generally do not translate proteins due to the lack of 5' cap structure for translation initiation, some circRNAs can be internally modified by M6A and start translating small peptides. The small peptide can well be bioactive, for example, as competitive inhibitors. Increasingly, circRNAs are found to interact with signaling molecules, such as AKT and PDK, to modulate signal transduction. One interesting circRNA function is acting as DNA sensor switch to control secondary messenger cyclic GAMP. In the nucleus, intronic circRNAs are tethered to the RNA polymerase II and enhance the transcription. Nuclear circRNAs can also tether spliceosomes for splicing or target chromatin modifiers, for example, TET1, TAT1, and DNA methyltransferase to epigenetically regulate gene expression. A classic circRNA function is microRNA sponge. SOR7 is well known for its massive number of 73 microRNA 7 binding sites. The other well-known example is circRNA SRY in the testes. It has 16 microRNA binding sites. These circRNAs bind microRNAs, AGO, and block target gene silencing. circRNAs acting as microRNA sponges are not just special cases. In fact, more than 100 circRNAs profiled by Arraystar circRNA services have experimentally confirmed the microRNA sponge functions. Increasingly, circRNAs are found aberrantly expressed in or causing diseases such as cancer, cardiovascular, neurological, metabolic, or immune diseases. circRNAs do not have free open termini, are resistant to nuclease degradation, and more stable in easily accessible biofluids. Compared with their linear RNA counterparts, circRNAs often have much higher cell, tissue, and disease specificity. These properties are desirable for biomarker applications. In blood, circRNAs can be present in cell-free serum, plasma, exosomes as cell-free circRNAs, or present in leukocytes or red blood cells as circRNAs, all of which can be used as biomarker for diagnosis, prognosis, guide clinical decisions, and monitor disease or therapeutic progress in cancer, neurological, cardiovascular, immune, and infectious diseases. Exosomal circRNA biomarkers can be further taken up by cells doing biological functions. For example, exosomal circRNA DB, SHKBP1, UHRF1, and HIPK3 can enter cells to act as a microRNA sponge or protein decoy to regulate ubiquitination, DNA damage regulation, angiogenesis, immune response, and cancer. In a survey of biomarker performance matrix, some circRNAs have shown very good sensitivity at a max of 0.86 and specificity at a max of 0.90. Here is a snapshot of many circRNA biomarker studies in the pipelines for disease indications. circRNAs can be epitranscriptomically modified by chemical groups such as M6A. Epitranscriptomic modification of circRNA is an important layer of gene regulation. As in mRNA, M6A, the methyl group modification at the 6 positions of adenosine, is the most important internal epitranscriptomic modifications of circRNAs. M6A is installed by writers, for example, metal 3, metal 14. The modification is reversible by erasers such as FTO and alkyl-BH5. M6A interacts with various readers to exert regulatory functions such as YTHDC1 for RNA splicing, nuclear to cytoplasmic RNA export, YTHDF2 or DS3 for RNA decay, IGFBP for RNA stabilization, and YTHDF1, 2, 3 for mRNA translation. As shown in this M6A immuno dot plot, M6A is the most abundant modification in circRNAs. The M6A bound fraction represents a larger proportion of total input circRNA. Among the circRNAs made out of a single long exon are modified most frequently, even much more than the unmodified circRNA, colored blue. Along the RNA length, M6A tends to be localized towards the upstream of the circRNA, whereas M6A is concentrated near the stop codon region in linear mRNAs. The M6A modification pattern in the expressed circRNAs shows substantial cell type specificity in this Venn diagram. Functionally, M6A modification in circRNA can be read by YTHDF3, which recruits translation pre-initiation complex to translate peptides even without a 5' cap structure required for mRNA translation. M6A can also be read by a different reader, YTHDF2, which recruits heat-responsive protein or HRSP12 and RNAsP complex. So the M6A marked circRNA is cleaved open for degradation. In the nucleus, an M6A modified circRNA can be read by YTHTC1, which facilitates its nuclear to cytoplasmic export. The exported cytoplasmic circRNA enzyme 2 is read by IGF-2BP2 to stabilize target HMG2 mRNA. In this model of circRNA induction or interferon response in innate immunity, a modified circRNA interacts with RIG1 in the presence of K63 ubiquitin substrate to induce MIVS filamentation, which triggers IRF3 dimerization and interferon production. M6A modified circRNAs is recognized by YTHDF2 reader, which repairs RIG1. So circRNA M6A modification shuts down interferon production. We have now come to appreciate the biological and clinical importance of circRNAs. An essential step to study circRNA is to profile their expression. The fundamental difficulty of using RNA sequencing for circRNA profiling is the lower abundance of circular junction sequences for sequencing coverage. On average, circRNAs as a population are expressed at about 1/10 of the average mRNA level. After the read sequences of a circRNA, only the circular junction spanning fragments are counted for circRNAs, which is even less at about 1/40 of the average mRNA level. The sequencing does not tell other regions of the body sequences coming from linear or circular RNA. In fact, most circRNA studies with deep RNA sequencing coverage could only see a few circular junction reads. A couple reads are only sufficient for detecting the presence of circular RNA, but not nearly enough to quantify its abundance, let alone quantitative differential expression analyses. Numerically, the sequencing recounts and the quantification error margin follow negative binomial distribution as shown in the orange curve. mRNA abundance at a higher level is reasonably okay with acceptable error margin. With the no abundance of circular junction read counts, the quantification error is so high to be acceptable. By using spiking reference, the limit of quantification, or LQ, that is, the minimum counts necessary for circular RNA junction quantification, must be greater than 300 counts. If you want to compare circular RNA differential expression between two groups, the limit of detections of ratio, or LDR, is 890 counts for a greater than twofold change. Very few circular RNA has these abundance levels above the limitations. In the data points from an actual experiment, RNA sequencing quantification accuracy is shown with the black curve, which is okay for medium and high abundance mRNAs. However, for low abundance RNA sequencing error becomes skyrocketing. In contrast, the microarray quantification accuracy, as shown in red, is relatively unaffected for the lower abundance RNAs. In fact, by sequencing, even the same RNA sample with different sequencing library construction methods and circular RNA analysis algorithms show very low concordance and large discrepancies, as shown in these Venn diagrams. A much better way to profile circRNA expression is by microarray. The circular junction is detected by unique probe sequence at extremely high affinity. To increase the sensitivity even higher, the RNA sample is treated with RNA-seq to remove linear RNAs but keep the circular RNAs. The sensitivity is high as 1 circular RNA per cell. The data processing and analysis are straightforward, without relying on special complicated circular junction mapping algorithms as RNA sequencing. The circular RNAs are analyzed with differential expression, annotated with genomic information, predicted for microRNA sponge function, such as microRNA binding seed regions, local AEO, and binding positions. The Arraystar circRNA microarray covers about 14,000 cataloged human, mouse, or rat circRNAs collected from circRNA studies and databases. circRNA epitranscriptomic modification profiling is actually a lot more challenging by mirror sequencing. Mirror has all the difficulties as circRNA expression profiling by RNA sequencing, as we discussed earlier. On top of that, mirror sequencing requires massive amounts of input total RNAs of greater than 100 micrograms. The profiling also lacks the modification stoichiometry, a key piece of information in epitranscriptomic profiling, as illustrated in the next slide. There are two key quantities or M6A modifications of each circular RNA. One is the abundance of the M6A modified circRNA. The other is the relative proportions of modified versus unmodified RNA molecules for the circRNA, which is referred to as modification stoichiometry or modification percentage. For example, 20% of our circRNA is increased to 80% modification upon drug treatment. Both measurements are essential pieces of epitranscriptomic information. The practical solution is Arraystar Epitranscriptomic Microarrays. The circular RNAs are immunoprecipitated by M6 antibody and further enriched for circular RNAs by RNA-seq treatment. They are modified circular RNAs in the supernatant not binding to the M6 antibody are labeled with cytidine 3 green color, whereas modified circRNA bound to M6 antibody are labeled with cytidine 5 red color. Both fractions are combined and hybridized to the same microarray so that the normalized intensities in two color channels are obtained. Both the M6A circular RNA abundance and the percentage of the modifications are calculated. So the microarray can provide the information of what circular RNAs are modified, stoichiometry of the percentage of the modified versus unmodified circular RNAs, differential modification between groups or conditions, coverage of about 14,000 cataloged circular RNAs, low RNA abundant, low RNA amount requirement at about only 3 micrograms. There is no need for polyase selections or ribosomal RNA removal as required by RNA sequencing. The microarray data come with easy-to-use analyses and annotation for biologists and clinicians. In this screenshot, the circular RNA differential expression is analyzed and annotated for statistical significance, p-values, fold change, FC, normalized abundance, percentage of modification or stoichiometry, circRNA type, small peptide coding potential, microRNA binding sites for microRNA sponge, and genomic information. As a roadmap for research forward, the differentially expressed circRNAs can be validated by circRNA qPCR. This confirmed differentially expressed circRNAs can be used for diagnostic or prognostic biomarkers, even without knowing their biological functions. To study the biological functions, overexpression for gain of function or lockout for loss of function are commonly used strategies. The circRNA binding proteins can be detected by comprehensive identification of RNA binding proteins by mass spectrometry or ChIP-MS, microRNA sponge function by competing endogenous RNA or ceRNA analyses, or small peptide encoding by mass spectrometry. For identifying circRNA binding proteins, you can try circRNA interactome to instantly predict proteins that interact with their circRNA. In comprehensive identification of RNA binding proteins by mass spectrometry or ChIP-MS, the circRNA in the protein complex is cross-linked and pulled down by biotinylated oligo beads. The pulled down proteins are identified by mass spectrometry. Conversely, circRNAs not bound to a protein can be identified by RNA immunoprecipitation or RIP assay. To analyze the microRNA sponge functions of circRNAs, competing endogenous RNA or ceRNA analyses can be used. These include bioinformatics prediction of microRNA binding by target scan miRNA or MutaMe, ChIP microRNA qPCR, where the ChIP proportion is the same as ChIP-MS as in the previous slide, but the microRNAs are isolated from the pull-down complex and detected or confirmed by qPCR as sponged microRNAs. The target mRNAs can be grafted to reporter gene so that the up or down regulations of circRNA can be correlated to the target mRNA activity. Likewise, for circRNA epitranscriptomic profiling, the differentially methylated circRNAs can be validated by MeRip-seq qPCR for the intact circRNA. To narrow down the M6A site in the circRNA, fragmented MeRip-seq qPCR can be used. The functions of M6A modification in circRNA can be studied by M6A consensus site mutation, gain of function or loss of function on M6A writers, readers, and erasers. To study the mechanism of action of M6A modification, you can look into the effects of M6A modification on microRNA sponge function by ceRNA analysis, AGO co-immunoprecipitation or reporter assay for the target mRNA. The protein binding by ChIP, RNA immunoprecipitation, RIP, circRNA stability by qPCR, and M6A initiated small peptide translation by mass spectrometry. Arraystar circRNA microarrays have been powering circRNA research in many areas of biological and clinical sciences, becoming the leading technological platform for phenomenal research successes. More than 400 papers have been published using Arraystar circRNA microarrays. Here is a select list of high-impact papers in last year alone. By profiling circRNA dysregulation on Arraystar microarray, circRNA ACTM4 was identified as a top differentially expressed circRNA in cholangiocarcinoma on this volcano plot. qPCR validation confirmed the upregulation in tumor. Overexpressions of recombinant circRNA ACTM4 promoted cancer cell migration and invasion in transwell assays. circRNA ACTM4 overexpression increases the metastasized lung cancer nodule, the number and masses, whereas lockdown decreases them in tumor in lung metastasis model. High circRNA ACTM4 level are associated with much poorer cancer prognosis and much sooner post-surgery cancer recurrence. Furthermore, circRNA ACTM4 can pull down YBX1 protein. Likewise, YBX1 can immunoprecipitate or rip circRNA ACTM4, demonstrating circRNA ACTM4 and YBX1 binding. Clinically, YBX1 expression is much higher in the cancer than in the adjacent tissues. By analysis of circRNA ACTM4 upregulated genes and YBX1 activated gene promoters, Freezo7 and LRP1 are the two genes in the overlap. In addition to the protein interactions, circACTM4 also acts as a microRNA sponge. 12 microRNAs were predicted as both circRNA ACTM4 and YBX1 RNA targets by searching circBase and StarBase. MicroRNA 424-5P is upregulated with circRNA ACTM4 lockdown. The base pairing of microRNA 424-5P with circRNA ACTM4 and with YAP1 mRNA are shown here, along with non-interactive mutants. Armed with the wealth of information, the circRNA ACTM4 molecular mechanism in Wnt, beta-catenin, and Hippo-YAP pathways is constructed. That is, in the nuclease, circRNA ACTM4 recruits YBX1 to activate Freezo7 promoter. Freezo7 is a freezed Wnt receptor in the Wnt beta-catenin pathway to activate YAP1 in the Hippo pathway. In the cytoplasm, circRNA ACTM4 acts as a microRNA 424-5P sponge, also targeting YAP1 mRNA. The activations of Hippo pathway causes the cancer growth and metastases. In another molecular cancer paper, circRNA circACTM4 binds FUBP1 protein, which then activates MYC oncoprotein activation pathway and promotes breast cancer. There were many more circRNA discoveries last year alone by Arraystar circRNA microarray customers. Published in Molecular Cancer, circRNA circIPO11 complexes with top1 protein to target and activate transcription factor glial gene promoter, consequently activating hedgehog pathway and driving hepatocellular carcinoma. In another molecular cancer paper, circRNA 220 acts as microRNA 30 sponge to target MYC oncogene like MYSM1 and immediate early response IER2 genes, causing acute myeloid leukemia relapse. Last but not least, circRNA-seq MIMO1 acts as microRNA 106B5P sponge to target TET gene, TET gene in DNA epigenetic demethylation, regulating epithelial to mesenchymal transition EMT pathway via TCF21, leading to lung cancer metastases and chemotherapy sensitivity. I will stop here for now. Thank you for your interest. Please visit us at arraystar.com.